Hello, everyone, and welcome to today's webinar, Tubulin or Not Tubulin, Heading Towards Total Protein Staining as a Loading Control in Immunological Protein Analytical Methods. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Protein Simple, a biotechny brand. To learn more, visit proteinsimple.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. This webinar is educational and thus offers free continuing education credits. Please click on the continuing education window at the bottom of your screen to obtain your credits. I'd like to now welcome our speaker, Dr. Christian Moritz, researcher, Institut Neuromyogène, Lyon. Dr. Moritz, you may now begin your presentation.
Thank you very much for the kind introduction. My name is Christian Moritz. I'm at the University and the Hospital of Lyon and Saint-Etienne, as was mentioned already. And I'm very happy to have this chance today to speak to all of you, with the title Tubulin or Not Tubulin, which this Shakespearean idea of this title came originally from my colleague Thomas Schulenborg, who I will also shortly mention in one of the next slides. And yes, the subtitle is Heading Toward Total Protein Staining as a Loading Control in Immunological Protein Analytical Methods. Main focus, Western blot, of course. So at presentations, me, for example, I always like to know where a speaker is currently sitting or is coming from. And so I'm currently sitting, I go back once, in this building here, and this building is located in Saint-Étienne in France. And that's where I'm currently doing my postdoc, and also Lyon, which is a city next to Saint-Étienne. And it's a nice little city. It's not the most beautiful city of France, but the ratio of quality of life and also costs is quite nice here. And I come originally, I did my PhD in Germany, which is here, where you see it, in Kaiserslautern. The cities are quite similar. Both are working class cities, which nice people, perfect to live. So, and also here in Kaiserslautern, during my PhD thesis in Germany, it was where I like to say where the Western blot part of my career began, because my very first paper, it's already some years ago, 2013, was a methodological paper about Western blot. And I still remember asking my boss at that time, Eckhard Friauf, what he would think about doing this methodological paper, try to test another loading control instead of the common ones like tubulin or so. And he was a bit skeptical about method papers, and he said, how long would that take to finish? And I said, I know which methods I want to do. It would be fast. I think we could do this all in 2 weeks. And of course, you all know how it works. Things last longer than you estimate at the beginning, and it lasted longer. But nevertheless, I never regretted to have asked him, and I was very happy that he agreed, although it took longer at the end, because it became an important part of my career. I would say I like to go into the details of a method, and yes, it even ended up today in giving this presentation, which would not have happened if I wouldn't have gone this first step into some method improvements. So, but I do not want to start with this publication. I would like to start a little bit more slowly. Why not with a fun fact you might have seen, or you might remember 2 years ago, 2019, there was the 40th birthday of Western blotting. And I had the chance to write a scientific birthday toast in the Journal of Proteomics. And I was also creating in the top left this nice or funny birthday cake with the candles to the 40th birthday. And it was quite funny after this publication. The impact, so not the standard, the classical impact factor, but more the social media impact factor, was quite impressive and even higher than for any of my other research publications, although I would say the others are more important from a scientific view. But here, the social media impact was quite nice, probably also because of this beautiful cake that I made for Western Blot. And I want to show you some comments. So what were the comments of the social media community? It was the Journal of Biological Chemistry here on the left who made a tweet, which was even retweeted 91 times. And while retweeting other people, colleagues, said a bit their opinion about the Western Blot and also congratulated to the birthday, like here on the very right, Sarah Courts. You see she's describing the love-hate relationship, or in the middle panel, someone is posting a meme of a standard Western Blot protocol, according to him, which always ends in a crying person. So maybe you find yourself in some of these descriptions. So I think, yeah, it is a cool method. We all love it and hate it at the same time. And in the 40th birthday paper, I was starting with a quick look into history, and I would like to do the same. I like history and I like science, so why not combining the two? So a quick look into the publications. The actual birth of Western Blot was in July, I think it was the 1st July 1979, where this publication here appeared from the group of George Stark at the west coast of the United States, California. And what they did for the first time was the transfer of proteins, which is the actual plotting step, from a gel to a paper, to a membrane, and then a detection with antisera. Which is interesting, maybe not a fun fact, is that the same group just 2 years before also described the Northern Blot, which was a first time playing with the words after Edwin Southern had invented the DNA plot and called it Southern Blot, or it was called Southern Blot according to the name of the inventor. And then the RNA blot was called Northern Blot, and then the Western Blot, which was not mentioned here yet in this moment. Western Blot, this was 2 years later. But only 2 weeks later, no, it was a bit more, some weeks later, here in September 79, there was another group, and according to the authors, they did not even know from each other. It was long before Twitter, it was long before the pre-publications databases. And so it's Harry Taubin et al., and you might know this name because he used a buffer, the transfer buffer that is still used and cited often. And here, what is different is again a transfer, but here it's for the first time an electrophoretic transfer. He uses SDS-URA for the polyacrylamide gel electrophoresis. He uses for the first time nitrocellulose membrane, and also primary and secondary antibody for the protein detection. Then 2 years later, you see, as I wrote in a paper, quite quite modern for the 80s, Western Blot has more than 2 parents. So the third parent, it's actually the part of parent who gave the name to the baby, was Neil Burnett in 81, who used for the first time, at least in a published version, according to the authors, they used the word already before, especially as they came from the West Coast, and so the word Western Blot was created. And I mentioned already citations. Funnily, the very first publication is cited least of the 3, but still an impressive number, more than 1,000 times. Taubin et al. Was cited most of all, more than 60,000 times. Incredible, probably also because of his buffer is still used today and often cited because of that. And Burnett paper giving the name almost 10,000 times. So I would like to go on with some, not history, now come back today, some more facts. And the question is, Western blots are outdated. Sometimes we hear this. I hear it, I'm also a part of the proteomics and mass spec community. And quite often in this community, I hear people saying, Western blot are outdated, and some are complaining that reviewers are asking after mass spec data to validate the findings with Western blots, and they say why Western blots is less exact as mass spectrometry. I do not want to follow this discussion here. This is another topic. But here are the questions if Western blots are actually outdated, and at least I tried to answer this question regarding the use. And so, of course, you could make a poll, go to different labs, see how often they use it. This is difficult, but we have the publications. We can count how often it is mentioned among different publications. And this is what I did here. So here I counted the proportion of publications mentioning Western blot in a text word. This is here on the top, the panels that you see here, normalized to all publications in the protein field. And you see that in the 80s, beginning of the 80s, there's a strong increase of this proportion, and then it reached somehow a steady state in panel A around 1990 until today, where Western blot is used in 8 to 10% of all studies, of all papers of the protein field. And this is much higher than other protein-related techniques like mass spectrometry in B, ELISA in C, immunohistochemistry in D, or microscopy in E. And so I would conclude, based on this data, that Western blot is still the most used protein analytical technique on the world. However, also this I would like to admit, very probably, although the most used technique, it has disappeared from the spotlight, because as you see in panel F, the peak when you do the same analysis, but now counting how often Western blot was mentioned in the title, then the peak was around in the 90s, around 1990, and then it went back to quite low numbers. So it happens quite rarely that Western blot is, the term Western blot is mentioned in the title, which is different for mass spectrometry, for example. It's more modern, more fancy to use mass spectrometry. So here it goes up to 6 per mil this time, not percent. So although it went out of the focus, the spotlight, it's still the most used technique, Western blot. And as you subscribed for this webinar, you all might have done already Western blots, know how it works, but nevertheless, I want to spend at least this first slide, one slide, to sum up main steps. So, and you can also try to find differences if you have used already the technique of protein simple. Our host, you can check where are differences between the classical Western blot and protein simple. You always need, I think, independent of the method, you need a protein source, of course, here in step 1. Then you need a protein extraction. You should determine the concentration. This would be a part of the extraction step here to see how much you extracted. Then in step 3, it would be a protein separation. Here's a difference between classical Western blot and protein simple, because protein simple is using capillaries. Also the transfer, step 4, is not done in protein simple, but in the classical version it is done. So you transfer the protein to a membrane. And then 5, it's a matter of discussion if this is still a Western blot by definition or already the next step, the detection of antigens. And there you can use either, as we do it in our group, often we use serum from patients, for example, to find autoantibodies. But most people of you in the audience might use commercial and purified antibodies in order to make bands, protein bands visible. So one of the first important question is, why actually do we need a loading control? And I want to show this to you, to explain it in a simple way. Is imagine here what we usually see in publications, in your results probably. You might compare different conditions. So let's say we have a control and a treatment. And our hypothesis, and this is what we also see here, is that the level of our protein of interest increases upon the treatment. But if you have only these information, a skeptical researcher should also always be careful and say, is this really the case? Did our protein of interest increase upon the treatment? Or can it not be that we just loaded much more protein here on the right side in the treatment lanes? To prove that you actually have loaded the same amount of total protein in all of your lanes, you need some control. Usually it is done with housekeeping proteins like tubulin or actin, for example. And this band, this tubulin or actin, this housekeeping protein, is something like a proxy or something like a marker for the total protein that you loaded into the lane. You see here on the left side now what you usually do not see, because you loaded a lysate of cells or lysate of tissues, for example, which consists of many proteins. Usually you do not see them, but they are there. So here you see that you have loaded more or less the same quantity of total protein of your lysate protein concentration into all of the lanes. So, and yes, and you use the housekeeping protein as a proxy, as an indicator for this total protein. So in this case, it's the optimal case. We do not have difference in our level of protein of interest, but we do not have difference in our total loading. In the next example, we could see what the skeptical scientist was maybe also expecting, devil's advocate. So here we see that the nice difference in our protein of interest that we saw, and was maybe according to our hypothesis, is actually not really convincing, because also the total protein, as you see on the left side, is much less. So we just loaded into the control, we loaded much less sample than in the treatment. And this has caused the difference in our protein level. We nicely see it on our proxy, on our housekeeping protein, which shows what we have loaded actually. But imagine now, or let's continue with the question, where does actually unequal loading come from? So here I simplified some of the steps that could be possible sources of unequal loading. And the first one is one of the first steps of your protocol. Usually it is the determination of the concentration of protein. Imagine you have your 4 samples, 2 replicates of the control and 2 replicates of a treatment, and it's 5, 5, and 5 microgram per microliter. But on the very right sample, you find 9, so a much higher concentration, which of course leads to the situation that you load less protein to have the same amount of total protein loaded. This is the goal. But in reality, imagine there was a mistake in your BC assay or Bradford or whatever you did. In reality, it was also 5, but you thought it was 9. So what you did is you loaded much less because you thought it's a high concentration. So if you have issues already in your concentration determination step, you will have an unequal loading on your gel. Another source of unequal loading is unequal sample pipetting, where you lose sample. I think most of you remember their first Western blot. I do, and I even more do remember students, internship students in our lab who did their first Western blot. And what happened very commonly is you load your sample, you go into the lane and put the very often bluish liquid into the lane. And then at the very end, there's a bubble and the tip of the cone is still too deep in the solution and the bubble rises and pushes out half of your soup. So you lose probably half of your sample. And this would be, especially for beginners, a source of unequal loading. Then the next source refers to the transfer. So far we were on the gel level. Now the transfer, so we transfer by electrotransfer, for example, the proteins from the gel to the membrane. So now we are having our bands on the membrane. And imagine there was, for example, a big bubble between gel and transfer, or for some other reasons the electrical field was not homogeneous, and now what is resulting is that the transfer on the right side of the treatment side was less efficient, maybe even in one specific part here in the middle part. And then in the worst case, imagine your protein of interest has issues in the transfer, but your housekeeping protein maybe is much lower and does not show a difference. So if you do housekeeping protein immunostaining, it will look nice and you will see a significant difference in your signal of interest, but in reality this was a technical issue and you will not see it when you use a housekeeping protein, but you would see it if it was somehow possible to make visible what you were actually loading and transferring on your membrane. Very similar, last possible source, I think there are more, but I mentioned here only 4, is if you do stripping and want to reprobe your membrane. Also, while stripping, you always lose not only the antibodies, but also sometimes proteins, and this loss of proteins can also be inhomogeneous, and then it results in the same effect as for inhomogeneous transfer. So this would not be, it's called source of unequal loading. It has the same, it's not actually the loading that is here the problem, it's a transfer, but it has the same consequences. So I think from this we all agree that we need something like a loading control. The question is rather, the discussion is, what should it be? And the traditional solution is housekeeping proteins like tubulin, because the general assumption is that they are equally expressed. This is what you read quite often, that they are ubiquitous, that they are at the same level, independent if you compare young against old, or, I don't know, green leaves of Arabidopsis against flower leaves. Yes, so this is the theory, equally expressed, but unfortunately this theory is not true. So housekeeping proteins are not equally expressed. I could give one lecture only about examples of in research of people, of colleagues, seeing in which cases they found housekeeping proteins unexpectedly to show a difference in two conditions that were compared. I do not do that. I show only one publication that convinced me when I was still in my studies, end of my studies here from 2007, 2008. It's colleagues from Czech Republic and United States, and they found, they called it déjà vu in proteomics, so already seen, and they published the hit parade of repeatedly identified differentially expressed proteins. So they checked different studies from proteomics using mass spectrometry, for example, where groups have compared anything, old against young, brain region A against brain region B, and so on. So anything that they were not very strict in selecting. They only wanted to know which proteins do people find. And then they found this déjà vu, these proteins that they strangely had already seen before in the paper before, who analyzed something very different. And so the list, I show you here the top 15 of the most identified differentially expressed proteins. Here the table for the families grouped together. And you see that actins and tubulins, depending if you take humans on the left, or studies that only address rodents on the right side. Here between 20 and 30% of all studies, of all comparisons between two conditions, found actins and or tubulins being significantly differentially expressed. So this means that these families are not at all equally expressed, which has an strong impact for the choice of loading controls for Western blots. So another concern, another issue with using housekeeping proteins is that they are often highly abundant, which is quite nice to have very clear signals, but it's also a disadvantage because here an image, I don't show the source because it's not the goal to blame someone. I don't even know it. I think here this I got from a colleague from Twitter who did an analysis of a saturation analysis, and he saw here on the right side that there is, we call it a ceiling effect. So usually the center of a band should be more intensive than the periphery, so it should be more like a dome. But here what you see is that the center and the periphery is very flat, which means, yeah, it's a saturation. There's a ceiling effect. And the consequence, unfortunately, is that if you would load now a bit more or a bit less, would not result in differences if you are saturated, if your signal is already saturated. So which scenarios could result from these issues? So we have already talked about this best case scenario. You have a difference in your protein of interest, but no difference in your loading control. This is something what you want to see. But imagine the second case, also this we have already seen. We think to have a difference in our protein of interest in red between control and treatment, but in reality, we just did not load the same amount of sample in the control versus treatment lanes. So now it can get more complicated, but usually these cases are more realistic. So here you see that unfortunately we are in one of these 20 to 30% of cases where your housekeeping protein is regulated, shows a difference. And if this by chance goes just in the other direction as your protein of interest, then unfortunately you will still have, this is the same scenario as in the slide before. On the left side, you see that in the controls, we loaded much less than in the treatment by any accident, by any reason. But we do not see it if the housekeeping protein on the right side in blue is either saturated, that could be one problem, or it is regulated just in the opposite direction. Yes, then you would publish a very nice, significant difference, which is actually, that you see on the left, based on a technical issue that you would have seen if you were able to see what you loaded, if you were able to see the total protein on your membrane. Last scenario, although this happens quite often, I read it in the internet often or hear it from colleagues, you have a perfect scenario on the left side, what you often do not see, the total protein. You see all is fine. You loaded the same amounts in control and treatment, but your housekeeping protein on the right that you chose shows is regulated in the same direction as your protein of interest. Maybe they even belong to the same family, or don't know. So they might be, in this case, they are regulated in the same difference. So you see, according to your hypothesis, probably a difference in your protein of interest. However, your housekeeping protein is not equal, and this you cannot publish. So your boss or the reviewer or yourself, in the best case, will say, no, there's something strange. I cannot show this. So these are not cases that I invented. They really happen, and in internet you find hundreds of questions in scientific discussion forums. Here I want to show you 6 questions, only the questions, not possible answers, just to show you what is the real world, from ResearchGate in this case. Here, first one, what are options for membrane fraction loading control proteins for Western blot? And he or she, they don't want to use sodium potassium ATPase for any reason. Maybe it's on the same level as of their protein of interest. And these questions, you can just exchange the word, the object. So what is a good loading control for? And then you find questions like Arabidopsis or brain or dinosaur bones or something like this. So you find the weirdest cases. Here in the next one, someone is searching for the good loading control in secreted proteins to the media. And then the third one, maybe it's my favorite because this person is so close to a solution, and it's actually a good example of our last scenario on my slide before. So what's a reasonable loading control, they are asking, for nuclear protein and acute injury? And now look here on the third row, on the second, how to explain, there's a typo, I think, that my total protein of a specific signal pathway is almost the same. So all is fine. He or she, they did a total protein staining. They are almost the same. But now the housekeeping protein, the histone, varies from each other in treatment and control, for example. So this is one of these cases where all is fine, but you think you have a problem because it's one of these 20-30% of cases where the housekeeping protein is just significantly regulated. So you cannot use this housekeeping protein. But this is the cool thing, so close to the solution, because they did already apparently a total protein staining and saw that the total protein, they loaded the same. So they had a loading control. They saw they loaded the same, but they didn't realize that it's a very good loading control that they had, which was not the housekeeping protein. Then, which are the best loading controls for Western blot excluding cytoskeleton ones? Which antibody should be used for loading control in Western blot when cells are getting bigger? Good point. She's writing, or he, they, I'm afraid that I'm normalized with tubulin, something else. And if the cell is getting bigger after treatment, maybe also the housekeeping proteins are getting more. This is a very good point, a realistic fear, and they are close to the solution. Last point, is beta-actin a good loading control in immunoblot for apoptosis studies? No, is it used? Yeah, probably it's used. It's another question if it's a good idea to use it. So the crucial question is, and this is important to understand, that what the community is currently using is housekeeping proteins that are a proxy for what we really loaded on the shell, for all proteins, for the total proteins on the left side that you hear. That is what you want to see. You want to have an equal loading of the sample that you loaded. So the question is, why going this extra way, and why not just directly making visible this total protein on your membrane? Then you see what you load and do not need some representative proxy, a housekeeping protein that might be completely off and might be not representative anymore for what you really have loaded, as we have seen in the examples. And this has been done already. So I did not invent that. I was not the first person who suggested just staining the total protein on your membrane. The only thing what I did 3-4 years ago was to collect these few publications that have already tested the total protein staining and used and tested in Western blot as a total protein, as a loading control. And so far, 7 total protein stainings have been used. You see them here: Ponceau S, stain-free techniques, Cyper-Ruby, epicocconone-based stainings, Comasi, Amidoplaque, and Cy5. For all of them, there is at least one publication that uses and shows benefits or parameters when they used it as a loading control in Western blot. And so I wrote a review publication about this, summarizing main parameters. And I'm quite happy that this publication is appreciated. So you see, I'm far away from Burnett's or Taubin's thousands of publications, but it's also not that long ago. Yeah, so I have around 20 citations per year, which is clearly above the average of this journal where this was published. And so I'm quite happy, and this happens unfortunately rarely to a scientist, that you publish something and then you see that really people use it and it has a positive, has a benefit for the community. This is, yeah, it's always a nice feedback. And so in this publication, in this review, you find many data which are, I do not want to go to this data. It would be a relatively boring presentation if I read you the dynamic ranges and the detection limits of each of the staining for each of the publications. This is something which is not feasible or doesn't make sense for a presentation, because it really strongly depends on what you are doing in your lab, which sample, which material you are working with. Maybe you have very precious material and can load only 1 microgram per lane, and then you have to choose one that is quite sensitive, or you have other restrictions. And that's why I made this huge table in this review to show for all of these 7 published total protein stainings, what are the parameters when you use them as a loading control in Western blot. And you see it here. First one is for Ponceau. You see it was the different publications. That's why in the first column you see the dynamic range, which goes from 10 to 45, and another one 5 to 120. So it also strongly depends on what the lab, maybe the membrane, maybe the material you use. And so I do not go into these details. I only want that you understand the different parameters and how and why they are important for the choice, for the selection. So we have the dynamic range, we have a detection limit, we have a technical variation, and I want to spend one slide about the variation, because this is something that we tested for one of the total protein stainings, the epicocconone. It's a fungi-based compound, and just to show you what it means here, biological variation or technical variation. On the left side, you see biological variation. You see that we did on the same blot, we tested the total protein staining on the top, then in A2, beta-tubulin and GAP-DH, and we tested this over 4 biological tissues, 4 organs, actually. And you see, it's even not that surprising, but it's still conflicting with the hypothesis of equally expressed housekeeping proteins. In A2, you see that the housekeeping proteins are quite different in the organs, while the total protein staining, although the pattern is also differing, but if you take if you really want to see what you loaded and take the whole lane, then the variation between the organs is significantly less, A3 panel, than for the housekeeping proteins. Same for technical variation. So here it's not different organs, but just different lanes to see the technical variation over the lanes. And also here, the total protein staining has benefits compared to the housekeeping protein immunostainings. Then the other points we saw in the table is dynamic range and a detection limit. And so the point is, if there is a loading difference, our loading control should be able to detect it, of course. Sounds simple, but it's not always that clear, because imagine this example is clear. You might have your difference that we spoke about before, but when your membrane, if your membrane after the detection of the signal looks like this means maybe you had only 1 copy of protein in the control and 5 copies of the protein of interest in treatment, then you still have 5 times more. You have an increase, a clear difference, but if you don't see it because 5 copies of a molecule are not enough to make it visible, then you are below the detection limit. Same in the other direction. When you load too much, you are saturated. We talked about that before, and you also will have issues in analyzing your data. So in reality, the optimal situation should be somewhere in between these 2 extreme cases, and this is what the dynamic range is. So you see it here again, simplified, 7 lanes on the left side. Same for the right side, should be the same thing, only on the left side it's a total protein staining, on the right side it's an example for housekeeping protein. Both of them have a detection limit, which is here in this case between 1 and 2. At 2 we have 2 microgram loading amount. We still have faint bands, but nothing in 1, which means our detection limit is 2 microgram. And on the other extreme, between 6 and 7, or we start between 5 and 6, we still have an increase in the signal, but if you load more, if you load 7 microgram instead of 6, you loaded more protein, but you don't have more signal. So this means you have a ceiling effect, you reached the limit, which is, and between 2 and 6, you have the dynamic range. Here in this example, they would be the same for total protein staining and housekeeping protein, but in reality, many of the cited publications in the table show that both detection limit and also dynamic range are much better for total protein staining than for housekeeping proteins. So I go back to the tables, although I don't go into detail. So now then you understand, if you go through the table and want to choose which total protein staining fits best for you, you know what it means. So I did this here for Ponceau staining. That was too fast. For Cyber-Ruby, for Coma-C, for Amido Black, for Stain-Free, for Epicoquinone, and last but not least for Cy5. Some, you see it here, for this example, they tested, for example, a dynamic range, but not a detection limit. That's why there's no information. So there's still many missing information. So something to do for you, for us, for the community. Yeah. So what I said, I don't go into details of this table, but I'm a more visual person. So here is some main results of the table summarized. It's all studies above the 7 total protein stainings that did analysis regarding the dynamic range. And you see here for Ponceau S, for example, there are 4 different studies. They are all a bit in the same range. We are showing some that the dynamic range is around 10, sometimes until 100 micrograms of total protein. And when you see at the extremities that the arrowhead, that means that the authors did not go to the limits. So they did not, here for example, of Ponceau S, reference 21, also 43, it is, I think it was logarithmic, so 5 microgram and there was still a signal. And when they went lower, no, they didn't test it. So 5, where there was still a signal, but they didn't try to find the limits, which was different for cyperubia and coma-C, for example. Here there's no arrowhead, there's the end of the line, reference 40. And here you see that 1 microgram of total protein was the last step that still was visible. If you load less, then you lose the signal, so you have a detection limit. And this can be helpful if you want to load very small amounts. Then you should choose your total protein staining accordingly and wisely, or this you see here in the blue box, or test other ranges if they have not been tested yet. The same applies for housekeeping proteins. So if you insist in doing housekeeping proteins, then even then you should test and even publish it, showing that you are still in the dynamic range, that it's not saturated, and yeah, that all is fine. So there are still some proteins, total protein stainings, that are maybe in the pipeline. So these are possible total protein stainings that could be used as loading controls. You see here a list of known and accepted total protein stainings, but they have not yet been tested as a loading control in Western blot. That's why they are not in the same table as the other candidates. But there's still space. You see, you can make a new publication. We could make a cooperation if we find the time and test some of these candidates as a novel loading control. At the end, I would like to refer to the protein simple system, because also here you can use the advantages of total protein staining. The protein simple system, we have quickly talked already in a slide before. It is quite close to Western blot. I don't know the current definition, if you, the audience, would like to call it a nice simplified way of Western blot, or if it's another category for you. It's like more semantic discussion. It's also not very interesting how we call it, but actually the steps are quite close to it, because you have the loading. You load your sample. This is an important step that you also have in traditional Western blots. You separate the protein, you immobilize it, and then you have the detection with antibodies. In the very left column, you can use in this case, a horseradish peroxidase labeled secondary antibodies. You can use chemiluminescence signal. You can use antibodies that are that have a fluorophore in the secondary antibody. Then you can use the fluorescence to detect the signal. You can, and this is on the right column here, also replex. Here the first part is the same as in the middle. Just what you do at the end is you can do stripping, can get rid of the antibodies before, and can add another antibody. Or what you can also do is to use a total protein staining. And this has already been done and tested for the protein simple system. You see here an internal example of protein simple. In B, you see the quantification of A of the left side, but the interesting part is actually the panel A, where you see some antibody against, it's a phosphorylation specific antibody, and this is a quite nice example, because you have actually 2 loading controls. You could even use the pan-X antibody as a loading control, because you see the second and the fourth lane, they are not empty, which you could be devil's advocate and say, if you see only these nice signals in the first and third column. But you see first from the red signal, but also from the total protein signal, that you really have loaded the same amount of sample. And the last example, I think it's my favorite, because this is how a total protein loading control should be tested for, because here on the top left, you see that what was done here is you really provoked some loading differences. You see here different loading difference. On the very right, it's 0.2, and then on the very left, it's 20 times less. You see it in blue in the total protein staining, but you see it also in black in your signal of interest. And imagine now there was an issue in the loading, and you loaded much less, but this is not a problem if you normalize, if you arithmetically normalize your signal with what you have actually loaded, and this is done in the panel top right. In the orange bars, you see normalized. And then even if you load different total amounts by accident or by intention, as it here was done here, then it is not a big problem, because if you normalize with what you actually have loaded, then you do not see a difference. And if you take only the raw data as shown in the blue bars, then you see, of course, clear differences. And if you do this by accident, you see differences that do not reflect your biological question, but more a technical issue. So this is the end, and I want to end again as I started with like a fun fact. And last week when me received the invitation, I saw this in the subject, and I copied it into my presentation because it is something that we can see what we learned. It can be one of the take-home messages, because I think it's not loading control versus total protein staining. I hope this is something what you learned from this talk, is that the total protein staining can be an example for a loading control. It can even be a better loading control than our classical loading controls. And another fun fact at the end, if you want to and still insist in using housekeeping proteins as loading controls, I'm fine with it. I cannot change it. And but maybe you have the same reason for keeping housekeeping proteins, like this colleague here. He copied this sentence from Twitter. The loading control can truly be the best supporting actor in the Western blotting world. Sometimes when gazing at a blot that leaves me filled with frustration and confusion, all I can do is walk away and say, at least the actine is beautiful. So I think this is maybe one of the reasons to keep housekeeping proteins, but scientifically not the best. So I hope what you learned, some main take-home messages, was that Western blotting is not outdated in any form, so neither in the traditional form nor in some perfectionized version of protein simple, for example. Then housekeeping proteins are neither locally nor temporally equally stably expressed. So not, at least not necessarily. There are examples of course, where they are, but in too many cases, they are not equally expressed. And total protein stainings as loading control in Western blot has multiple advantages over housekeeping proteins. So open for questions. Thank you for listening to me. And yes, I'm looking forward to discussing these topics now with you in the question-answer session. Thank you very much.
Thank you, Dr. Moritz, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. So let's get started. Our first question is, would staining with tubulin be an accurate way to prove loading quality when trying to measure protein expressed into conditioned media?
I think, this sounds like one of these very specific questions, one of these examples that I just told you from ResearchGate. So I understand the question as, what if I do research in proteins that are released to the medium and I want to make Western blots with the medium to find there some protein of interest. So can I use tubulin? And it's a good example because it shows that we do not know. So we must find out, is tubulin really released to the medium? In the first moment, I would say no, maybe not. So it would not be a good idea. But it shows that we don't know. So either we would need to do the extra work and find it out and look, is it strong enough, the signal, and is it stable? Is it equally expressed between the conditions? Or that would be the alternative, to just use one of the total protein stainings as an alternative.
All right, thank you. Next question. What about staining the spent gel rather than the membrane?
Yeah, that's a good question. I addressed this point in my review article, but I didn't mention it now in my presentation. This is something that is also sometimes recommended. And you remember my slide where I was talking about the possible sources of unequal loading, and doing the protein loading control total protein staining on the level of the gel would control for the first two sources, which was the issue in the concentration determination, and also the loading itself. That we would perfectly control with a total protein staining on the gel level, but we would not see any issues that come through the transfer or later by stripping, for example. So it helps, but not everything. So I prefer the total protein staining on the membrane level because it covers also the transfer. That is an important step.
All right, thank you. Our next attendee first says, awesome talk, thank you. And their question is, for inhomogeneous transfer, how would you address that issue?
Yeah, so I understand this question, so not how to control it or to find it. So then the next step, if we see the unequal transfer, so if one student would ask me that, I would say repeat the Western blot, and if it's continuously the right side less transfer than the left side, for example, then there might be some issues in the setting. Maybe then you should check all the cables or something like this, because I think this should not come through the buffer, because if everything is fine, this should be equal. Yeah, so this is really difficult to answer because it could be so much. So I would just repeat it. Maybe you have a second setup or try it in another floor with a neighbor group. Then you can rule out if it is the material or rather the buffer or something like this.
All right, thank you. Next question. May I know why I see big differences in total protein signal across different organ or tissue, even I had to quantify and loaded equal amount of protein?
Yeah, this would be surprising to me. I also did something similar and even was provoking seeing differences by checking different organs. And what I showed on one slide was that the pattern is indeed very different. So some bands are very strong in one organ, but not in the other. But then when I checked for the volume, the total signal over the lane, that was quite similar. So that should be the case. So it's not a problem if the pattern is different. This is normal between organs, but the intensity should be the same. If this is not the same, then yeah, either there's an issue with the quantification, the Bradford or something like this, that for some organ-specific proteins it works better or worse, or in some organs maybe highly abundant or hydrophobic proteins are overrepresented, and then they might be lost during the transfer. So these could be reasons why you see differences even in the total protein staining, that the numbers are not representative, more, as I said, hydrophobic proteins. That could be an explanation.
Okay, thank you. It looks like we have time for one more question. What if a reviewer asks me to use a classical housekeeping protein as a loading control?
Mhm. Yeah, I think that happens quite often. I hear it also from colleagues. And so what I usually say is the reviewer is a scientist, and we scientists should be best convinced by data. So what one could do is just to cite one of the publications showing that this type of total protein staining is a good one, or yes, or the review article to show that it is more and more getting standard to use total protein staining. And yeah, that would be the way I would try to convince the reviewer.
All right, thank you, Dr. Moritz. Do you have any final comments for our audience?
Just thank you for listening, and yeah, it was a pleasure for me. And yeah, sorry that so few time was left for the questions, but feel free to contact me. And I think that questions that have not been responded and that come later, I will receive by email, and I will be happy to get in contact with you. Thank you for listening.
Well, thank you again, Dr. Moritz, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Protein Simple, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.